Hello and welcome dear viewers to the another video on bioequivalence basics for generic product development. See, for the generic product filing and regulatory approval, along with the other things like stability studies, validation studies, and the other requirements, bioequivalence is most important and is a must requirement for generic product. So for regulatory approval, the bioequivalence is required for the products like tablets, capsules, and many other products, excluding the solution type of formulations, or for the formulations for which the BCS bioware can be taken or can be granted by the regulatory authorities. For the all other formulations, whether these are IR formulation, extended release formulation, modified release formulations, this bioequivalence is required. So bioequivalence becomes the key parameter for the generic product development. And in this video, we are going to study the bioequivalence requirements and the parameters or key considerations for the bioequivalence studies. So let's start with the video. See, the generic products are developed for the cost effectiveness, and nowadays the regulatory authorities are also promoting the generic drug development, and the government bodies are also promoting for the generic product development. Why? Because these generic products are cost effective for the patients. And why these are cost effective? If you consider that, why these generic products are cost effective? Generally, the generic products are those formulations which are replica or which are like the innovator formulations, but these generic formulations doesn't include the preclinical and clinical studies. Only the bioequivalence studies are performed for generic formulations. These are the non-patented type of formulations many times, or these are come into the market after the patent expiry. That's why these formulations are cost effective. For getting the generic drug approval, the generic applicant or manufacturer is required to demonstrate the bioequivalence. And bioequivalence is nothing but the similarity in the rate of absorption and the extent of absorption with the reference formulation. Sometimes it is also called as RLD or reference listed drug formulation, specifically for US Europe market, or it is also known as market product or reference formulation or the comparator product. Now, what is bioequivalence? The rate and extent of the absorption of the test product, like generic product, do not show a significant difference from the rate and extent of absorption of the reference product, you can say, or compared to the reference product when administered at the same molar dose under similar experimental conditions in either a single dose or a multiple dose studies. So that time you can say that the bioequivalence is achieved. Now, in simple way, the rate of absorption from the test formulation and the rate of absorption from the reference formulation is similar, and also the extent of absorption is similar. That time you can say that the generic product is bioequivalent to the reference product, or you can say that the bioequivalence is achieved and demonstrated. The main parameter is that the dose should be similar and the experimental conditions should be similar when performing the bioequivalence studies. Now, this similarity gives you the similarity of pharmacokinetic profile, and based on this pharmacokinetic profile similarity, the therapeutic similarity can be demonstrated, or safety and efficacy can be demonstrated with the reference formulation. So this is regarding the bioequivalence definition or meaning of the bioequivalence. Now coming to the bioequivalence basics, like 100 mg dose of the comparator product and the test formulation is given to the patient, and then the plasma concentration profile is made. So you might have seen this type of plasma concentration profile. Here you have the Cmax. Cmax is the maximum concentration that is achieved, and the time at which this Cmax is achieved is known as Tmax. So this is Tmax and this is Cmax, and now this area is known as AUC. So the rate of absorption is demonstrated by Cmax and the extent of absorption is demonstrated by the AUC. So always remember that Cmax and AUC are the key parameters for the bioequivalence studies. So this is regarding the single dose bioequivalence study, and this is regarding the multiple dose bioequivalence study. Here you have started giving the dose, then at this time you have given another dose, so you keep on giving the doses, and after 4 to 5 half lives the steady state is achieved. I have made specific video on steady state bioequivalence. You can go through that video to understand this terminology in detail. So this is the figure for steady state pharmacokinetic studies, and this is the figure for single dose study. Then what are the key considerations? Whenever you are performing the bioequivalence study, you need the to be marketed prototype formula and the product that is manufactured with the finalized manufacturing process or intended manufacturing process with all the excipients and GMP conditions, and you require the reference product. Then you require the study protocol. So that protocol will give you the clear understanding and the written steps for performing the bioequivalence study. Now see here, we will see the key considerations for bioequivalence study. These are as below: type of study, study design, strength to be investigated, subjects, analytes to be measured, bioequivalence basis, waiver request of in vivo testing, dissolution requirements, first point Cmax, endogenous compounds. And another thing is that sometimes BCS bioequivalence can be taken, and also sometimes based on the formulation design, directly product can be given a waiver from bioequivalence study. Like if the product is in solution form or gas form, that time you are not required to perform the bioequivalence study. So type of study may be fasting state, fed state, or the steady state. So fasting state means the drug is given on empty stomach, and that condition is known to be more discriminatory because food effect is not there, and the true difference between the reference formulation and the test formulation can be demonstrated by fasting state. That's why many of the formulations are tested or are studied for the bioequivalence study in fasting condition. Then fed state. This fed state studies the effect of food. Many of the times the formulation is studied in fasting as well as in fed state to demonstrate that the formulation behaves similar to the reference formulation in both the fasting and the fed state. Generally, fed state is performed when the innovator gives the label claim that take this tablet or take this product on or after meals. So that time fed study is required. If the formulation of innovator claims that in their labeling or PIL you can say that there is no food effect, so that time generally the study is performed on the fasting state. Then coming to the steady state. Steady state is multiple dose study, and in this study generally the modified release formulations are tested. Some of the formulations which are toxic or which cannot be tested into the normal human beings, that time these formulations are tested in the patients who are already taking those medicines. Then many of the times the drugs which can get accumulated and the PK profile can be studied only after the rate of absorption and the rate of elimination is constant, that means the steady state. So for those formulation or for those drugs which shows the accumulation kinetics or which get distributed inside the tissues with a longer half life, for elimination for those formulations and drugs steady state type of bioequivalence study is done. Then coming to the study design. So you might have seen that single dose, two-way, crossover, in vivo, or replicate, full replicate, partial replicate. You might have heard these things. So these are very simple to understand. See, single dose means the patient or subject will take single dose of innovator and single dose of the test formulation. That's why it is single dose. If the study is multiple dose, then it will go to steady state or multiple dose studies. Then crossover. Crossover means if you have 20 number of subjects, you can divide those into A and B. 10 will make group A and 10 will make group B. So first A will receive the test formulation, same time the B will receive the reference formulation, and then it will be crossover. So B will get the test, and in the next time A will get the reference product, or vice versa. TRRT, like this you can understand. So this crossover study may be replicate or non-replicate. Non-replicate means only once you have to give the test formulation and the reference formulation. But if it is replicated, that time the reference formulation can be given 2 times, that is known as partial replicate. And if the test formulation and reference formulation both are given twice, that time it is known as full replicate. Then coming to the single dose two treatment randomized parallel study. Parallel means A will receive the innovator or a reference formulation and B will receive the test formulation at a same time, and this will not be crossover. So that is the parallel study. Multiple dose studies. In this multiple dose studies, these are done in crossover design fashion or in a study design of crossover. So many formulations are there which require multiple dose studies. So these are also known as steady state design. Then strength to be investigated. So generally the higher strength is the strength for bioequivalence, but the strength which is more critical for demonstrating the bioequivalence, that strength is selected. The other strength may be lower or higher based on the safety and analysis requirements. So generally higher strength is taken for bioequivalence study. Other strength, middle strength or lower strength can be taken based on the safety and analysis quantification requirements. If the higher strength is not safe, that time the middle or lower strength can be taken and the other strength can be waived from the BE study. Bracketing can also be possible. If the formulation is having five different strengths, that time the lowest and highest can be taken, or highest and middle can be taken, and based on that the other strength can be given the biowaiver. So waiver can be given to the other strength which are other than the bioequivalence strength. Then subjects. Subjects are the human subjects, and these subjects are selected depending on to the drug properties and the formulation design. Generally, the number of subjects taken for any B study is based on the statistical power, and statistical power is nothing but it is the significant data. If the drug substance is variable and if number of subjects are taken very less, that time whatever the data you will get, that will not give you significant information, or it will not have that much statistical power. So the number of subjects are selected based on to the formulation design and the drug molecule properties. Generally healthy subjects, both male and females of both are taken. Patients are taken as subjects when there are safety concerns, and sometimes the subjects are selected considering the specific requirement for the drug product, and the subject selection is also based on the metabolism properties. If the formulation is getting metabolized in liver with some specific pathway, and if that metabolism is very significant, that time the subjects are selected based on some liver function test or kidney function test, so that there will not be any bias in the data. So this is regarding the subjects. Sometimes the only female subjects can be taken, sometimes only male subjects can be taken. Their age, weight, BMI, everything is noted down during the start of the B study. Sometimes only female subjects are taken. For example, if the study is for some female hormones, or if study is for only male hormones, so that time the subjects are taken based on to the mechanism of action of the drug or therapeutic indication of the medicine. Now coming to the analyte to measure. See, whenever there is a intake of medicine or formulation by oral route, the drug goes through metabolic pathway, and the drug get absorbed, distributed, metabolized and eliminated. So if the drug goes under significant metabolism, and if there is less possibility of finding the parent molecule in the plasma, that time metabolites are required to be studied. So generally the appropriate biological fluid is taken like plasma, sometimes or rarely it is urine, and in that the analytes are measured. Generally the parent metabolite is measured, and sometimes the metabolites are measured. Rarely, both the parent and metabolites are required to be measured based on the metabolism pathways and the effect of metabolism onto the drug pharmacokinetics and drug pharmacodynamics. Now coming to the bioequivalence criteria. So as we know, the 90% CI limit for AUC and Cmax is 80 to 125, and this 80 to 125 limit can be widened for some highly variable drugs for Cmax. See, the average BA should be within the 20% range of the reference product. That means from 80 to 120 it will go, but it is not like that. It is having some statistical calculations, and based on that it will go from 80 to 125. The confidence interval of 90% of the relative mean of AUC of the test product should be within the 80 to 125 of the BE range. That is within 80 to 125 of the reference product. So this is the BE criteria. Now waiver request for in vivo testing, that is bioequivalence criteria. So acceptable bioequivalence studies on the BE strength. If you have a product with multiple strengths, that time one or two strengths will be BE strength, and based on that bioequivalence demonstration, the other strength can get the BE waiver. But the other strength are required to be similar in dissolution. So acceptable in vitro dissolution testing of all the strengths is required, and dissolution similarity is required to be demonstrated with the strength which is passing into the BE study. For example, you have three strengths like 100 mg, 50 mg and 25 mg. That time you have done, for example, you can say 100 mg strength you have studied in the bioequivalence study, and it is bioequivalent with the reference product. Now your 50 mg strength and 25 mg strength should demonstrate the dissolution similarity with the 100 mg strength, which you have proved that it is bioequivalent. Then proportional similarity of the formulations across all the strengths. So this proportional similarity is another criteria. For this I have made 2-3 videos, and the title of those videos is Bioequivalence for additional strength. So you can go through those videos to have in-depth understanding. Then are the resolution requirements. Many of the times the formulations require multimedia dissolution other than the QC release media. For example, if your QC release media is water, that time you will require to have dissolution to be performed in 0.1 normal HCl, 4.5 acetate buffer and 6.8 phosphate buffer, and other media also depending on the formulation. Then dissolution similarity with the B strength is to be demonstrated, and the criteria is F2 criteria or bootstrapping method or MSD method is used. Then alcohol dose dumping test by dissolution testing. This is generally required for the modified release formulations, prolonged release formulations, sustained release formulation, and some of the delayed release formulations. For alcohol dose dumping also I have made some videos. You can please visit those videos to understand better. So alcohol dose dumping study is required, and it is required to be demonstrated that the test formulation is behaving similarly or behaving as similar to the reference formulation in the alcohol dose dumping dissolution studies. Then coming to the first point, Cmax and endogenous compounds. So first point, Cmax means if you take the samples from the subjects, that means blood samples, and you observe that on the first sample you are getting the Cmax. That means there is some error in the sampling times. That's why the first sample is showing the Cmax. this happens for the drug formulations which are very rapidly absorbed. So when the first point of concentration time curve is the highest point, maybe due to some improper sampling time, and this can be improved by increasing the sampling time or sampling frequency near to Tmax. So whatever the Tmax is there, nearer to that more sampling can be done, and a pilot B study can be performed to understand the Tmax time. Then endogenous compounds. So these are the compounds or drugs which are synthesized or which are present in the body, like some hormones are there. So levothyroxine sodium tablets, these are containing the levothyroxine sodium, and levothyroxine is also produced inside the body. That's why the baseline correction is required for levothyroxine during the B study, so that it is demonstrated that the blood plasma levels of the endogenous levothyroxine is not interfering with the bioequivalence. Then coming to the BCS based bioverse, see BCS class 1 and 2 are the BCS class 1 and 3 are the class of BCS which are highly soluble, and class 2 and 4 are low soluble. So BCS based biover can be taken for the formulations containing BCS class 1 and 3 drugs, and which are showing rapid dissolution with Q1 Q2 similarity. So I have made a video on BCS based biovers. There I have mentioned all the requirements in very much detail. Then other doses from specific biovers like solution form and gas form. If the formulation is available in solution form or gas form, that time the B study is not required to be performed. Many of the times nowadays Q1, Q2 and Q3 type of requirements are there, which are the requirements for Q1 means qualitatively similar composition to the innovator, Q2 is quantitatively similar, and Q3 means the formulation is similar, the drug formulation design is similar, and the API used is also the same. So these are the composition similarity requirements. See, some of the formulations are required to have BIND. That means it is required that whenever going for the bioequivalence studies for these formulations, you have to contact the regulatory authorities with complete data of in vitro testing and stability before initiating the bioequivalence. So BIND is generally required for the drug products which are not safe, and some of the products which contain the cytotoxic drugs. So for those BIND is required. So this is regarding the B study requirement, bioequivalence requirement for generic formulations, and I hope I have covered the basics and basic terminologies for B study. You can watch other videos which are already available on the Pharma Learning in-depth channel to have in-depth understanding and complete knowledge. Thank you for your time, and thank you for watching the video. Keep learning and keep watching the videos. Thank you.